Okay, it is Monday morning. Hope you had a good weekend. March 29th, we are on chapter 26 still, putting together amino acids. We made peptides last time. We'll get into proteins today. And hopefully finish it off. Quiz 8 and homework 11 are due tonight. And then we start sugars next. So chapter 27 will be sugars. One other thing about resources for this class, and this is the Organic Chemistry with Biological Applications book. It's a custom book we had put together by John McMurray. They made a mistake on the cover. It says Chem351. If you go over to the bookstore and try to buy this, it might be under 351. It's inexpensive. It's the biomolecules chapters with more detail. I recommend it if you're going on to other molecular biology type classes or biochemistry and want more fundamentals. Although my overheads and everything handout wise will suffice for when we do metabolism and the other biomolecule chapters coming up. So you don't need this. We don't assign problems from it or anything, but it is a good resource book. So you can go over there and take a look at it and buy it if you'd like to. All right, questions on anything? Yes, please, Reese. What's that? Will resolutions be on it? No. So you can read through that and see how amino acids are resolved, and you can separate the enantiomers. So the question, will resolutions be on the exam? No. It's a way to get a hold of the enantiopure amino acids. You recrystallize them with a chiral amine, and that's the basis of that. The two crystals are diastereotopic then. They have different properties. And it's similar to what we talked about with transition states for the asymmetric synthesis. Okay, but that's a good question. Very good. All right, let's go up here first to the overheads, Jesharon. And here we got reactions. We talked about these protecting groups. And remember, for synthesis here, you can just start with the protected amino acids. The one we start out on the C-terminus side is always an ester with a free amine. Then the next amino acid is always T-Bach protected, or we could just say Bach for our class. So we have the free amine, free carboxylic acid. DCC couples very quickly, and then TFA takes off the T-Bach to get ready for what? The next coupling. So we can just do those two-step sequences over and over again. And they've been rendered to react with very high yields. These react with 99% yields in minutes. Both of these reactions are very fast. So it can be automated, and it mainly comes down to this reagent, DCC, carbodiamide, which we talked about. You know, here's our free amine, carboxylic acid. The amine actually takes off the proton temporarily. You attack the carboxylate to this middle carbon, push the electrons over and transfer a hydrogen there. So I'm just showing the intermediate. It's got the urea, you could say, or uronium if you protonate this, with the acyl group there ready to go. So this is just a better, leaving group, right? And then the amine attacks, tetrahedral intermediate, collapse back down, and urea is your byproduct. Okay, that can be easily removed or filtered off, okay? So it can be automated. Bruce Merrifield at Rockefeller University in the 1960s made bradykinin, which took many years and many chemists to make. And it's just a small little 11-mer peptide, but he was able to make it in a couple days with just one person running the machine. And the key was really to immobilize the first amino acid on a solid support. And maybe you can't see this. My next graphic, I blow this up. You can see this a little better. So this is polystyrene resin, which is, of course, not soluble. It will swell a little bit in the solvent. You put the first amino acid on there. It's just an SN2 reaction. There's a primary benzyl chloride on the polystyrene. You put it on there, and then TFA or HCl takes off the Bach, and then you're ready to DCC couple the next one. Bach, take the Bach off, and keep going. And at the very end, then you have to hydrolyze some things, treat with stronger acid or base, ammonium hydroxide or sodium hydroxide, whatever. You have to take off the side chain protecting groups. That's usually hydrogen and palladium. And then you get your peptide. This one's a little bigger, and you can see the structure's better. GEEK, sorry about that, but we got a couple side chains we need to protect here. Our first amino acid is what? Lysine, K. And there it is with a CBZ on the side chain. And we have the benzyl ester here. Now, it could be the solid support. That could be the polystyrene polymer. Do you see a couple of the next one, which is glutamic acid, which also protected the side chains, is a benzyl ester. Notice that'll come off with the same deprotection step as the C terminus benzyl ester. Then the next one's just Bach off, put on another glutamic acid. And then what's our last one? Oh, glycine. That's a simple one. Bach protected glycine goes on, see no side chain there. And then hydrogenate, all those benzyl esters, CBZ comes off. It's a benzyl oxycarbonate. And then the Bach simply comes off with a little more acid, HCl, and you get the intact tetrapeptide in that case. Questions on that? Okay, let's go over here to the sideboard, Jesher. The next thing we need to talk about is blowing peptides apart. So kind of seems counterproductive, but we just talked about how to make them. Now, why would we want to destroy them or hydrolyze them? Well, a lot of times we have new peptides that are being discovered in cells by biochemists, or they're new proteins that are finally being expressed and purified from new genes found within cells. The human genome has about 25,000 different open reading frames, makes 25,000 different proteins, and we haven't determined all of them yet. That's still a very open, ongoing area of research. So people need to determine the structures of new proteins all the time. And here are the methods that are used to do it. Structure determination, the order of the amino acids. Well, we do it in a destructive manner. Sorry about that. We break them down to the point where we can analyze them easily. The first approach is kind of a very aggressive one, partial hydrolysis. We just treat with strong acid and usually heat, and this will take hours sometime, and we get small peptide fragments. We break it down and convert it into a bunch of smaller problems, okay? To determine the sequence of something that's hundreds of amino acids long directly is very difficult. We divide and conquer, you could say, right? The other way is we know a lot of proteases, or peptidases they're called. These are enzymes that are specific for cleaving polypeptide chains. They're cannibals. They are peptides, they're enzymes, they're globule balled up, and they have a binding site for specific regions of peptide substrates. And it can cleave on the C end where specific amino acids are found. So the first one's trypsin, next one's chymotrypsin and carboxypeptidase. These are digestive enzymes that are useful in the digestive tract to break down food proteins into smaller amino acids. Maybe you've heard of those, pepsin and other ones that work in the stomach. So these are well known. Trypsin will cleave on the C side of what? Arginine and lysine. And by the C side, I mean right here. So it'll liberate a peptide over here and over here, but the key thing it recognizes is the charged side chain, arginine and lysine. Then chymotrypsin is a little bit different. It recognizes what? The big aromatic amino acids, and it cleaves right there on the C side. So at phenylalanine, tryptophan, and tyrosine, okay? And right there. And there's one more the book points out, and that's carboxypeptidase, which only cleaves the amino acid on the C terminus end, and that's it. It does that one quickly, and you might think, well, can it just keep going? No, usually stop the reaction at a certain point and then just characterize whatever this amino acid comes off on the end. At least you'll know what the C terminus is. That's a little bit limited. These two in combination are very powerful, especially with the Edman degradation that I'll show you in a second. Sanger method's kind of an old one. It relies on nucleophilic aromatic substitution, which is a reaction we've actually seen. It involves this dinitrofluorobenzene. So the N terminus is a primary amine. And this can do the addition here, form the stable anion intermediate, and then kick back off the fluoride. And it puts this colored, this highly colored, it's a yellow material, the dinitrobenzene, on the N terminus. And then you chop up the amino acids and you see which one has that yellow color, okay? Or fluoresces, whatever. And that is just limited to the N terminus there. just like carboxypeptidase is limited to the C terminus one. So those are kind of limited. The Edman is really the workhorse. And this is the one where we take these small fragments now from the partial hydrolysis and do the full sequence analysis of the smaller peptides. And it is fully automated. It uses this phenyl isothiocyanate reagent to form a thiohydantoin intermediate. And you'll see what that is in a second. It'll be automated here, and you use mass spec determine all the specific amino acids. And they come off in sequence from the N terminus on to the C terminus of your small peptide. And then with the mass spec analysis, you know specifically what that amino acid is and what spot it was at. That's the most powerful technique, and it can be automated. There's a machine called a peptide sequencer, which is a fantastic thing, along with the peptide synthesizer. Those two things really have revolutionized the study of peptides in the lab. And then we have a more modern technique, and this is continuing to evolve too. This is multi-ion mass spectrometry, where we look at fragmentation happening from the different ends. It's a very high energy technique, but you can see the different peptide fragments and the molecular ions, and then lead up and do the sequence analysis. We're not going to go into that. We'll show you the Edman degradation, as it's called, sequencing technique, mainly with the partial hydrolysis and the trypsin chymotrypsin idea. Okay, let's look through this first and see how they work. Back up on the board here, Jeshurun. I'm making you go all over again. All right, here's the Sanger method with this pentapeptide. So again, it does the nucleophilic aromatic substitution right here on the N terminus. So whatever that primary amine is here, okay, that's the nucleophilic one. And then you get the dinitrophenylamine there. Okay, so the fluoride comes off, you see. And so that's nucleophilic aromatic substitution. So you can review that reaction. I think it was in chapter 19 a while ago. But, you know, the lone pairs of nucleophile come in. And then you have negative charges by the two nitros. You can kind of see pushing the electrons. I'm not showing the whole thing here. And then partial hydrolysis, or in this case, full hydrolysis. This would be many hours and heating it up in strong acid. And this is, you know, pH 1 or 2. So this is super strong acid. Eventually, amides will be hydrolyzed, right? They are derivatives of carboxylic acids, but it takes very vigorous, extreme conditions normally to get them to hydrolyze. Esters come off first, of course, okay? But eventually all these amides are cleaved here. You get the individual amino acids here. And you see they're just the free amino acids. You'd use electrophoresis to separate those. But here, this valine would have this yellow dinitrophenyl group on it. That would stay intact. You don't break this aromatic nitrogen carbon bond with these conditions here. You just break the amide bonds, you see. And you get this DNP or dinitrophenyl valine derivative. That at least tells you valine was on the end, okay? All right, Edman degradation. This is the biggie. And like I said, this can be automated in an instrument, a machine. You just put in your little peptide, and it can't, actually it can go up to 100 MERS now. They have very sensitive instruments that are very high yielding now. Up to 50 is pretty routine, okay? So if you chop up a new big protein into some smaller fragments, you can easily get the sequence analysis here in this case. So let's see. It's this phenyl isothiocyanate. There's a mouthful for you. There's a new functional group we haven't talked about before. It's the same idea. The nucleophilic one is the primary amine, which adds here to the central carbon, kind of like DCC. Okay, so the nucleophile adds there. Push electrons, transfer a proton over here, and you get this thiourea intermediate. Okay, you see here is our amine. It added right there, transfer a proton over here. And that's urea is the two amino groups, and then a carbonyl. But here it's to a sulfur double bond, so we call it a thiourea. Okay, well, that's not the end of the story. You see your amide here is in proximity to come over and form what? A 5-membered ring and a tetrahedral intermediate. That tetrahedral intermediate can break down, transfer the electrons over here. So I'm not showing the full mechanism. You can practice that. In fact, I think it's one of the homework assignments for homework 11 to go through the full Edman mechanism. But I'm showing you enough of it here. I think you can figure it out. Bring the electrons down, right? Push the carbon-nitrogen bond off. And then you get what? You get the more simple peptide with one less amino acid on the N-terminus end. And you get this thiazolone intermediate. Now, that's not the end of the story. Except this can enter into the machine and keep going. You can add another thing here after you filter off and get the thiazolone, which actually rearranges here to what's called the phenylthiohydantoin. Okay, and that's a thiourea with the ring here in place. Notice the thiazolone has just a sulfur thioester and the nitrogen here. So you have to do a rearrangement here. You have to open up the thioester. Thioesters are labile. Rotate around and have the nitrogen here on the phenyl attack over to the carbonyl. And you see, then you'll get the rearranged thing. This is actually the stable thing that forms after a few seconds. This is the one that's actually filtered off and goes into the mass spectrometer. We've talked about mass spectrometry. That's where we ionize it. Then based on the molecular weight, you can tell what amino acid that was because they're all unique there, right? So valine would have a molecular mass different than glycine, etc. So by the mass spectrometer that looks at the individual phenylthiohydantoins, you can tell what the sequence is because the first one coming off is the N-terminus, then the next one, then the next one, and it's automated, sequential that way. Your next one would be the smaller peptide. Here I'm showing it coming off. If this is glycine, you see, this is your thiohydantoin then with no side chain. Then your next one is what, arginine? That has a big guanidinium thing here. Then your last one would be what, leucine coming off, okay? But they all come off as those heterocyclic derivatives called hydantoins, which are thioureas really. You see the urea arrangement, but it's with a sulfur there. Okay, so that's Edman. Questions on Edman? That's the trickiest one, I think. It's got a tricky mechanism, but it's also the most useful, the most powerful. Okay. All right, what's the next one here? Already did Sanger. Okay. Oh, partial hydrolysis. And again, don't underestimate the power of this. This is kind of random, and we're doing a simple peptide here. But you could think of doing this with a long chain with 100 or so amino acids. And you just treat it for a limited amount of time with real strong HCl water, right? 6 normal. That's that's going to be very, very strong, okay? And it still takes hours to do this because we're breaking amide bonds, but we're getting the fragments. But within the fragments, all of the primary sequence is still maintained, okay? So they don't recombine under these conditions. So we're just partially hydrolyzing the main chain of the original peptide or protein, okay? So we get the individual fragments here. You can see this is VEGRL. So you see all of them here. Here's VEG maintained, right? That's just this one breaking there. You can see all these others are just fragments of that. So all that information is maintained. What goes on operational in the lab? These are separated by chromatography, and then each one of these smaller fragments is submitted to the Edman sequencing, okay? And then from that sequencing, we use computer programs to just slide them along and determine what the original sequence must have been. So it's kind of a molecular puzzle, if you will. You've got to figure this out. The simple ones we might have on a quiz or a test, you can just slide them along and see how they line up and see what the original structure may have been, okay? So here's, V, EG, and here's EG arginine, right? And so, glutamic acid glycine lining up in both of those are there, you see. And you see val-glu there, which also relates to that tripeptide. And then this arginine leucine relates to this tripeptide. And then the computer can just slide them along and see what's consistent with the final thing there. We can use this in conjunction with the carboxypeptidases, the proteases they're called. These are hydrolases. They use water to hydrolyze the amide bond. I'll show you the mechanism of one of them here on the board in a second. They're usually serine or aspartyl proteases, which are really fantastic because normally this takes hours in strong acid. But these proteases, trypsin, chymotrypsin, operate at room temperature under neutral conditions. And it's really a proximity geometry thing. They have a binding site for the peptide. They hold that peptide bond in its location. A lot of hydrogen bond goes on, and it directs water or a serine off the same active site of the protein directly toward the carbonyl. So it's the proximity and the geometry which lowers the transition state energy and provides a pathway that allows for the cleavage of an amide bond, which normally would take many hours in higher temperature and strong acid. So these are fantastic things, enzyme-wise, that are able to do a reaction that normally is very difficult. But there's a summary we just had on the board. I'll leave this up here. Those of you who are live and want to look at this, let's do an example here and see how we can figure this out. Let me give you a problem here. So let's take a 14-mer. What do we mean by that? Well, we've got different amino acids here. So C terminus is starting on the N terminus, and there's 14 of them here. And our goal here is to figure out the primary sequence or the order of those amino acids from the N to the C terminus N. Remember, we use that convention when we talk about peptides or proteins. The N terminus is always on the left, C terminus on the right. Of course, they can tumble around in space, whatever, but the connectivity is the important thing, right? The order there. Let's take first here chymotrypsin. And chymo is going to what? Cleave on the which side? Well, the large aromatic ones, phenylalanine, tyrosine, tryptophan. And let's see, when we take our 14-mer here, we'll get AKF, we'll get GDW, we'll get LH. And these little fragments are microsequence. That's how we get the value here. So we know the ordering of these fragments, okay, that came out of our chymo digest, we say. Okay, so what are the results here? Chymo cleaves where? Oh, it cleaved right there on that C terminus. So that's probably not the C terminus end. It cleaved right there, you see. What about this? Oh, there's tryptophan. Ah, look at this one. Histidine, is that the end? Is that the C terminus? It might be, okay? Because that's not a site where chymo would cleave, right? How about the other one? Ah, tyrosine. There it is. Okay, so we don't know the overall order of our 14-mer yet, but we already tell some localized order here on some specific fragments, right? Okay, let's take trypsin, which is our other protease. Again, it's an enzyme that does specific amide bond cleavage. And what's the data here? Well, we'll have AK, we'll have MVR, and we'll have FGDW, SR, and we'll have YLH. Okay. Now, where does trypsin cleave? Okay, well, it cleaves right here. Arginine, okay, on the C terminus. K, lysine. Remember, that's lysine. That also has the charged ammonium cation. Both of these are the basic large charged one. That's why the binding site of trypsin, it recognizes those. Again, on the C side there. And then what do we have here? Oh, another arginine. So cleavage must have. Oh, and there's our H again. Okay, so is that the C terminus? Yeah, at least our data is consistent with both of those. Okay. So this is a simple enough structure. I think we could start to link this together, right? So you can already see some things here that are lining up, right? Let's go through this and see here if we can just reorder some of these things. Let's take this one here, SR. And how about that one? Look, just transpose it down here. Here we got SR, SR overlapping, right? So maybe M, V, R, Y just continues there. Oh, and there's our Y here, Y, L, H. So we've got some overlap there and there. So the computer does this for you. But with simple enough ones, I think you can see how to do this. And what is that it? 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12. No, what else do we need here? Ah, we've got AKF right there, right? AKF. And then we can, propose here a structure, and then we can make sure it's consistent, right? So is it AKF GDW SR MBR YLH? Okay. That is indeed a 14-mer. Is it consistent with all our data? We can go back and double check, chymo, yeah, cleaved right there, and there, yeah. Oh, and there, yeah. And where did trypsin cleave? Well, it cleaved right there, and the two R's right there and there. Yeah, and that would be consistent with the data. That'd be enough to say that we've determined what the primary sequence of that peptide. Okay, questions on that? See how that went? It's a molecular puzzle, you could say. Again, you got to make sure it's consistent with the data. There's some simpler examples, I think, in the homework assignment, so make sure you look through those. Let's look at these protease mechanisms here. How do these molecular catalysts, really, trypsin and chymotrypsin and others, pepsin, we know quite a few of these, how are they able to do this at room temperature in minutes? Okay. Because normally, to cleave an amide bond, we have to use strong acid, heat, and hours to get it to go. Again, it's binding these things with a recognition element. It has a more complex structure. It holds it in proximity with the water. Okay, they are hydrolases. They break these bonds with water. So how do they do it? Well, we need to look at the mechanism. Okay, and I'm not going to show you the full structure. We'll look at some full structures here in a minute. We'll go back to Proteopedia. But let's look at a substrate here with an amide. And we've got a couple of the R groups here. And so that's just our substrate, we say, okay? So here's our amide bond that we're going to cleave, right? We've got R and R prime there. Okay, well, the enzyme here is a protease. And the enzyme has a protonated base. It has a proton source, now held in proximity to that. So here's our enzyme, okay? And it's this big thing here. It can be hundreds of amino acids, okay? And it has a binding site, that's kind of where this region is here, that can recognize this based on what that structure R group is, right? Whether it's arginine or lysine or whether it's one of the big aromatics, okay? What else does it have in proximity here? Well, it also has a serine. Well, at least trypsin and chymotrypsin are serine proteases. Some are aspartate, which have two protonated carboxylic acids, which actually transfer a proton to the carbonyl of the amide. And that's what we're going to do here, right? We're going to transfer this proton here and make the carbonyl more electrophilic. Well, what's also in this binding site is another base, okay? And usually these bases are histidines, lone pairs, okay, on the aromatic imidazole, which can take a proton off or donate a proton on, okay? So it's proximity and geometry here. How is this going to work? Well, we can imagine this base taking a proton off of serine. And this serine is part of the chain. When we say S here, here's our primary alcohol. It's part of the chain. And if we take that proton off, what can we do? Well, we can attack here and grab the proton off there. Okay. So I'm kind of doing a lot here, but what? We're hydrolyzing this, okay? Or we're going to break this bond. We don't see water yet, okay? We will use water here in a second. But that initial step here does what? It forms a tetrahedral intermediate, which has the serine on it, okay? Has the amine here, and it has the other R prime over there. Okay, is that a tetrahedral intermediate? Yeah, it sure is. There's our carbonyl of our amide. And again, it's due to the proximity. Okay, it's in close proximity to that serine, and it's being protonated. It's being made more electrophilic. We're getting to this point much more readily because we have everything lined up geometry-wise. Okay, what can go on now? Well, we've got this base right here, which has a proton on it now, right? And we've got this base up here, and this is all part of the enzyme, right? So this base can take that proton off. These electrons can come back down, and what? Can break the bond this way. This carbon-nitrogen bond is in closer proximity to that now. And what do we get? Well, we get the serine still on there, okay? And we get the free amine. So there's our cleavage of the amide bond, okay? Look, we just grabbed the proton back off our proton source here, and temporarily we have an ester. An ester is more reactive than an amide, right? Now the water actually comes in. Let's see, how does the water work? Well, we've got that base, and we've got water. So here's the water now. We can grab proton off water. That can come in and what? Form another tetrahedral intermediate. Okay, so there's a reason why we taught you all those reactions with tetrahedral intermediates. Well, they carry over into biology. And what are we going to get here? Well, you're going to get, yeah, it's okay just to have that as O minus. The serine can just be there. And then what we have, the protonated base still there. So any of these slashes here are just the enzyme still working there. So, well, the slash here is the rest of our substrate. Yeah, keep these two things straight. The enzyme's the catalyst, the big thing with all the groups coming in. The substrate is just our peptide. And we've already liberated part of it, right? Okay. But right now on the C side of that, we've got an ester, another tetrahedral intermediate. Yeah, that's okay. And then look, this can just come back down, grab the proton off there, and look, then we've got our carboxylic acid, our substrate, okay? And yeah, let's differentiate that very carefully. There's our substrate over there where our amine just came off. Here's our substrate here, and what about our catalyst? Where our catalyst, look, just went back to serine, okay? So here's our catalyst, or our enzyme. Okay, well, I think that's the full thing because now we've got our amide bond cleaved. We've got carboxylic acid on one side, got amine on the other. We've gone through two tetrahedral intermediates. Why? The first one was very stable, right? To get to this here, this is the slow step, okay? But once it gets here, this is a more active ester now. Esters are hydrolyzed more quickly, more rapidly. Again, it's proximity here. This brings in the water molecule and knocks that off. So it is a hydrolase. And that means you use water to cleave bonds. It's a very common family of enzymes. We'll see the rest of them here in a second. Question on the mechanism here for this protease. Like I said, there's also aspartate ones where there's two protonated carboxylic acids right in the binding site, and they actually donate a proton to the carbonyl. And they bring in water also. Maybe we'll leave that on a future quiz to look at. All right, let's look at the rest of the story here. I think Jesherin will spend quite a bit of time up here on some of the overhead material. So let's look at the full structures of proteins. So conformations, we got a chain here, okay? And N to C terminus, and a lot of freedom here. We can rotate around these bonds, but not around the amide bonds. Remember, we said those are planar and flat. But we've got alpha helices, different regions in the peptide chain, especially when they get over 50. They begin to adopt these localized structures. We got the coiled structure here, the alpha helix. Maybe you've remember that, or these extended open things which have a lot of hydrogen bonding across here. There's a lot of hydrogen bonding within the alpha helices. Well, how do they determine these structures? Like I said, there are 25,000 open reading frames in DNA, cDNA. And so all these different structures, how do they come about? Well, we need to summarize first a couple things on the board. So sorry, Jesherin. Let's go back to the board here. And we're going to summarize the basics of protein structure. We've already talked about one of the basic ideas, and that is sequence, okay? So the main thing, or the primary structure of a peptide chain or a protein is the sequence, okay? And we've already been talking about that, right? The order of the AAs, okay? And that's everything, really. If you can determine that, you can figure out the rest of the three-dimensional shapes of things. The secondary structures involve the alpha helices and the beta sheets, okay? And the turns, we should say. Now, the alpha helices are well defined. They're the coil type structures. And then the beta sheets are the extended zigzag type structures. The turns are kind of loop regions, and they're kind of undefined. They have a lot of conformational freedom, even in the folded up protein. But they're part of these secondary domain type structures. The tertiary structure here is the folding. And this is into the three-dimensional structure. So, you might have a couple of alpha helices, maybe a loop, another alpha helix, and then maybe some beta sheets, and then the end. Okay, it gets really messy here. But this is determined from the primary sequence. Now, how does it do that? How does it fold up? If it's a globular protein, it'll reliably fold up into this state after it comes off the ribosome. Hundreds of amino acids, and it'll find this right three-dimensional fold. Well, we'll have to look at that, okay? The amino acid, the side chains, okay, non-polar will be what? Inside, okay? The polar side chains will be out. And believe it or not, that's a big part of why things fold up the way they do, right? Because here you'll have what? The non-polar ones inside here, and you'll have the polar ones on the exterior, okay? And it folds up in such a way, because they're down the chain, at different spots. And this folding up turns out to be a very important thing. If you want to win a Nobel Prize, come up with a new way to calculate that. Go directly from a complex primary sequence and come up with a specific three-dimensional fold. Okay, still an unsolved problem. And then we got quaternary structures, and that's just multiple chains. Okay, and we'll see that with hemoglobin here in a second, which has multiple chains. So yeah, let's go back up here. Okay, so yeah, here's a big peptide chain. In fact, a couple of them held together. This is insulin. You see some disulfide linkages between cysteines. The 3D structure here, we got some cartoon structures. These are the alpha helices, these coils here. We got some loop regions, and you can see the disulfides here, the yellow balls are. Insulin's not an enzyme, it's not a catalyst, it's a hormone, it's a signaling molecule to tell cells to bring glucose into cells. Okay, it's a signaling molecule. You can just look at the sequence. There's an A and a B chain. They actually start out together, but there's a post-translational modification where they're hydrolyzed by a protease, a specific one, to turn it into the active hormone form. Here's a large complex enzyme. This is involved in the synthesis of tryptophan in cells. And it has 267 amino acids, and there they are drawn out. That would be the systematic IUPAC naming approach, where all of the prefix from the N terminus on, and only the C terminus end is named serine, not serial. Okay, wild names. I won't hold you to that, but thank goodness for simplified names. We call this tryptophan synthase A, okay, instead of naming this whole thing here, okay. So that's kind of a practical issue. Here's some secondary domain information, alpha helices. Some amino acids are more prone to be in alpha helix regions. These right here, I won't have you memorize that, okay? Just know the basic topology or the shapes of these secondary domains. So alpha helix is a coil structure, and it coils around to the right. And that coiling effect is due to the stereochemistry, the S stereochemistry at the alpha carbons, okay? And you can also hydrogen bond down the coil. And that hydrogen bonding helps stabilize that structure, okay? You can, of course, hydrogen bond to water, but if you adopt this topology, this shape, you get this beneficial internal hydrogen bonding network, okay? We abbreviate these as ribbon structures. You've already seen some of those, but you see the coiling? It's a right-handed helix or right-handed screw type shape. You can see some of the side chains here. Here's tryptophan, here's phenylalanine, here's another tryptophan, a valine. Okay, oftentimes we just emphasize the stripped away alpha helix. Here's the beta sheet or beta pleated sheet, just like skirts or pants can have pleats in them. Um, and it's this zigzag structure. Most of them are anti-parallel, where the C and the N termini line up opposite of each other, and then you get this regular intramolecular hydrogen bonding pattern between the two. Okay, so they're not coiled up; they're extended zigzag structures conformationally, and then they hydrogen bond across like this. If they're parallel, the hydrogen bonding arrangement's not quite linear, but there are certain amino acids that prefer beta pleated sheets. Okay, so those are localized domains within proteins. Okay. And then we got the cartoon view of the sheet. And you see this is flat, extended here, with a loop in between. And then these arrows are usually pointing from the N terminus to the C terminus. Okay, and maybe you've seen some of those. There are certain repeating motifs, like this Greek key motif, you see over and over again in certain proteins. And so, yeah, the whole thing, you know, here's the extended primary sequence. They eventually fold up into these localized structures, depending on what amino acids there. Proline will not be in either helical form or sheet region. Where did I say the prolines are usually found? Remember that? They don't fit nicely into the helix or the beta sheet. Where do we find the prolines here in this structure up here? Yeah, in the loop region, yeah. So these would be over here, and we'll look at some other diagrams here. So the tertiary structure is the folded up, the ball-shaped globular structure, and then aggregate if you've got multiple ones. Here's a nice summary of all of the effects within inside the chain. Hydrogen bonding's dominant. We've got nonpolar interactions, which are in the interior usually. Hydrogen bonding across the chains, or hydrogen bonding of the side chain to the main chain, or electrostatic interactions, okay? So we can have these salt bridges, are sometimes called, where we've got oppositely charged side chains together there. And we got hydrogen bonding here, nonpolar. Oh, and the disulfide. That's the only other covalent linkage within the amino acids. So the disulfide linkage here. And then alpha helix here with hydrogen bonding there. Well, can we denature this? Can we go from the folded state and go back to the unfolded primary? Yeah, we can do that. How do we denature or unfold proteins? And we're not breaking the amide bonds. When we say denature, what do we mean? We can do it with solvents, okay? Add a different type of solvent that will unfold different things, different hydrogen bonding. We can do it by changing the pH. That will change different salt bridges, right? pH will do it. What's another way to denature a protein or change the shape? And you see this all the time in the kitchen. When you put egg white in a pan and start to heat it up, what happened? It becomes white, right? What's that effect? You're actually not breaking the chicken albumin protein down. You're actually denaturing it. You're changing the folding state. Then it aggregates in a more efficient pattern, and that leads to the opaque color. So heat will also do it. Well, so what did we say? Solvent, pH, or heat can denature. A lot of times that's reversible. You just take away the heat or that solvent and it'll go back. A lot of times denaturing those is irreversible. So here are the main types of proteins. We've got fibers, collagen, which are these bundles of amino acid chains. It's mainly glycine and proline and what's called hydroxyproline. Now that's a post-translational modification where on the proline, remember the 5-membered ring there, proline's unique, there's an extra hydroxyl. And that requires a cofactor in the body. Anybody remember or know what cofactors required to make collagen, connective tissue in the body, synthetically? The cofactor is a what? A vitamin? Ascorbic acid? What's another name for ascorbic acid? Vitamin C, right? And so this pathway to take proline and make hydroxyproline. And why do you need hydroxyproline in collagen? Well, it forms another position for more hydrogen bonding within these coiled up chains. Now, these are the fibers that hold together connective tissue, right? Sinews and connective tissue between bones and muscle. So this is very important. If you don't have enough hydroxyproline, you get a disease called scurvy, where those degrade, and you have a lot of problems. So certain vitamins are critical, and one of them is vitamin C to make hydroxyproline. Anyway, those are kind of like helical structures. You got keratin, hair fibers here. And we got silk, which is a different type of protein fiber. It's mainly beta sheets, okay? But these are insoluble proteins, right? They're not dissolving. We need them there. Helical bundles or any globular type protein is more soluble. The external positions, we have the polar side chains, and these can bundle up with multiple alpha helices. You can see here, that's very common. And they can actually span lipid bilayer membranes that form channels for different ligands, different substrates that can go through, signaling molecules or hormones often need to be transported specifically through the membrane, and that's a recognition event through these proteins that can span the membrane there. And some of these are very complicated, these barrel type structures. Again, it's a control issue. Myoglobin is probably the most well-studied globular protein, 153 amino acids. A couple classes ago actually had this up on Proteopedia. We'll look at this now in a little more detail. So here's all the order of the amino acids from the N to the C terminus here. And it has a cofactor. It has this heme ring right here, which has iron in it, okay? Transports oxygen in muscle and heart tissue. It's actually prevalent in marine mammals, dolphins and whales. I think the one here comes from whales. Hemoglobin is in red blood cells, erythrocytes in the body, right? The red color is due to the heme rings, the iron that's there. There's two chains, two alpha chains and two beta chains. They actually have different sequences there, but about 150 amino acids. And then the four chains come together into a what? A quaternary type structure, okay? So the tertiary structure is one chain folding up, and then quaternary is multiple chains coming together. And it's a cooperative effect. Hemoglobin needs to bind oxygen in the lungs and then release it in the tissues. And that release is actually dictated by a small change in pH once it gets to its target spot. And this is a so-called tense versus relaxed state of hemoglobin. You'll learn more about that later. But what I want to point out here is a specific mutational form. If this valine right here, six amino acids in, is mutated, I'm sorry, if this aspartic, if this glutamic acid E is mutated to valine, you get the sickle cell anemia form, okay? And that's very prevalent in people of African American descent, or African descent, I should say. 10 to 15% of that population have this mutation, which is glutamic acid to valine there. And this is that sickling that can occur. So here's a normal erythrocyte. They're little donut-shaped cells. And if it has that mutation, if it's just that glutamic acid to valine, and that valine's non-polar now, that changes how it folds up. And actually, the hemoglobin will begin to aggregate, and it'll form these structures within the cells, which lead to the sickling effect that's actually seen. And those are more rigid, and they can't pass through the small capillaries in the body as efficiently. So they have circulation issues and breathing issues. And that can lead to a lot of other secondary diseases. But it's just due to one single mutation there. Now, there's a lot of other mutations that are known for different diseases. We're not going to get into that. But here's the porphyrin, here's the binding of oxygen. Again, we call that a cofactor. It's another molecule. It's not the amino acids, but it's part of what needs to be in there. A lot of times cofactors are derived from vitamins. We'll see that later. We're running out of time now, so we'll need to do a little bit more on this. Protein folding needs to be reliable. If not, you can lead to different diseases like Alzheimer's disease. These beta amyloid proteins don't fold up correctly. They can aggregate and form these fibril plaques in the brain, which lead to that disease. And we'll talk more about the next time, the 6 classes of amino acids. But we've already finished all the topics. I think we're fine for quiz 8 and for the homework to be due. So we'll just have a little intro when we start sugars on Wednesday to wrap up proteins. So I think that's good. Questions on anything? Just know the basics of this stuff. You don't need to know the specifics. We're not going to ask you, a lot about the diseases, sickle cell anemia, and things like that. But we're just showing you how the sequence analysis and things like that are important, and how the sequencing can occur and how it relates to the structures. Questions on anything? Very good, guys. Thanks for being here, and we'll see you on Wednesday. Okay.